Hello everyone, my name is Madhura Nayak and I will be your presenter today along with Dr. Mindy Wang, our Senior Scientist and Probe Design Lead. Today's webinar focuses on RNAscope Probes, specifically how to identify the right probe for your research. A few key points to note during this presentation. You will be muted during this presentation. Please use the chat feature available to you to ask your questions while this is in progress. If we cannot answer your question during this webinar, we will get back to you offline. Let's review the agenda for our webinar today. We will begin by reviewing our probe design technology basics, which will focus on the ZZ probe design. We will then review our probe classification and nomenclature, which will help us identify and differentiate the probe. The next topic on our agenda focuses on finding the right information on our website so you can determine the best possible probe design for your experimental target. Lastly, should our greater than 13,000 unique probes be insufficient to meet your target needs, we can design a custom probe for your target of interest. Mindy will discuss the requirements and information needed for us to provide a probe suitable for your study. Let's begin by reviewing our probe technology. RNAscope and BaseScope is based on proprietary in situ hybridization technology with unique probe design and signal amplification. This platform has the sensitivity to detect most genes in situ, as well as simultaneously quantify multiple RNA transcripts. Just as any in situ assay, RNAscope assay involves permeabilizing properly prepared samples to allow complementary oligo probes to bind to RNA target of interest. Specific signal is amplified using the double Z probe system, which is subsequently detected and quantified. The assay is extremely universal as well as rapid and scalable. To design a specific probe for your target of interest, ACD requests you to provide us either the accession number information or the exact sequence of the target. You could even indicate your region of interest for targeting with start and/or stop positions. We will elaborate it in the custom probe design session later in the webinar how you can get this information. Our bioinformatics scientists channel this information through the proprietary ACD alignment algorithm to select optimal sequences to ensure target specificity and uniformity of oligo hybridization in the assay. Our algorithm evaluates the sequence composition of each oligo in the pool to ensure no cross-hybridization with non-target RNA under stringent RNAscope conditions. So how does the technology work? RNAscope probe design is the key. We use what we like to call the double Z probe design. Now there are three components of the Z. The lower end region of the Z binds directly to mRNA. This lower region contains 18 to 25 base sequence, and that is specific to your target. A spacer region is in between the base and the other end, which allows flexibility for probe binding efficiency to mRNA. The upper region is the 14 base tail sequence, and that is the region where the preamplifier binds. The Zs are used in pairs. Both Zs of a ZZ pair bind adjacent to each other to your target region from the lower region. The two tail sequences of a ZZ pair form a 28 base hybridization site for the preamplifier binding and the cascade of amplification to occur. So typically a 10 kb region on the RNA molecule is targeted by about 20 probe pairs. Now the yellow line represents your RNA. If your RNA is intact, under ideal permeabilization conditions, the assay will hybridize up to 20 Z probe pairs to your RNA. But for many samples, such as cancer FFPE samples, your RNA could be degraded and fragmented. So the simplicity of this design and the relatively short probes binding to the target allows for binding to even short pieces of RNA. So how does the signal amplification work? You have your target transcript, the probes are added, and the double Z's land adjacent to each other. The pre-amplifier will then bind to the top region, which is the 28 bases, and each amplifier will then serially have 20 amplifiers bind to it. On each amplifier, there are theoretically 20 labeled probes 20 times. So we see a 20 Z pairs with 20 amplifiers and 20 labels, which is theoretically 8,000 labels per transcript. What about non-specific binding? So if a single Z binds to non-specific RNA, the pre-amplifiers cannot be stable on just the 14 bases of the upper region. So the non-specific binding that may occur does not get amplified. And that is how the double Z design prevents background noise and contributes to specificity. Based on this background technology, our RNAscope probes are suited to a wide variety of research applications, such as cancer, neuroscience, and infectious diseases, to name a few. The benefits of our RNAscope probes can further be elucidated by the availability of over 13,000 unique pre-designed probes available to select from. These probes are universal for virtually any gene, any species. They are specific and sensitive with a performance reliability. We also provide a 2-week turnaround time for custom designs. And our probes are ready-to-use formulations, which makes them easy to use. This wide use of our technology has also translated into over 730 peer-reviewed publications across many different journals. To identify the right probe for your experiments, let's now review the basic probe classification and nomenclature as it applies to RNAscope probes. Probes can primarily classify by the assay type, namely RNAscope probes, which are designed for targets that are greater than 300 base pairs, and basescope probes, which is specifically designed for targets shorter than 300 base pairs in length. RNAscope probes can also be classified by the platform used to run the assays. These can be manual or on the Ventana or the Leica autostainer. Lastly, our probes are classified by the channel that they are designed in. And this channel indicates the compatibility of the probes with different amplification systems. We will discuss this point in greater details in the following slides. Let's now look into our probe nomenclature and start by identifying the different probe types. As shown on this slide, the probe type is indicative of the platform the probe can be used on, either manual, which is denoted by the RNAscope probe, or Leica autostainer, as denoted by RNAscope 2.5 LS probe, or suitable for Ventana autostainer, as indicated by the RNAscope 2.5 VS probe. The next denomination in the probe nomenclature is the species. Species refers to the probe target species and implies that the probe is designed to the gene of the specified species. The species is typically identified by 2-letter designations such as HS for Homo sapiens, MM for Mus musculus, etc. Next is the official gene name, and this refers to the official gene symbol as listed in the ncbi database. The specification part of the gene name indicates any specific target region with the gene. Please note, this may not be available for all probes. If you notice the designation O, O1, or O2, it just refers to an alternative design to the same target gene, such as either a different start and stop region or a different number of probe pairs. The last denomination in the probe nomenclature refers to the channel. Channel segregation is required when multiplexing multiple probes on the same sample. RNAscope probes default to channel 1 unless specified. This channel indicates the compatibility of probes with different amplification systems and indicates which detection kits the probes would be compatible with. C1 probes are compatible with all detection platforms. C2 probes are compatible only with the 2.5 duplex detection kit and the fluorescent detection kit. C3 channel probes are only compatible with the fluorescent detection kit. If you plan to multiplex 3 probes using our multiplex detection kit, each of the 3 probes would need to be in a separate amplification channel. With this background knowledge of our probe classification and nomenclature, let's move on to identifying and accessing probe information from a catalog probes on our website. Please note, our complete list of catalog probes is available for download on our website and can be accessed through the highlighted link here. Please also note, a general guideline of the probe classification and nomenclature as discussed in our previous section is available as a helpful resource to navigate this extensive probe catalog. Our website is the most updated resource for catalog probes with 50 to hundreds of probes added to our catalog every few weeks. The quickest way to search for our catalog probes is by utilizing the search function on our website. You could choose any of the search parameters listed on this slide here, such as gene name, species, or specific information such as accession number related to the target sequence. Using the official gene symbol will result in the most accurate results. In the next few slides, I will demonstrate how to retrieve my probe of interest using human CD274 as an example. Please use the search button on the top right of the page to type out the gene name. In this example, we will use CD274 or PD-L1. This search resulted in 42 results, which now can be refined further by platform, species, and channel using the refine bar located on the left corner of your page. Using these filters, in this case, I refine my search for manual assay, species human, and probe in channel 1. This resulted in about 5 results. To further drill down and identify the target region of these specific probes, please click View Details and a drop-down table will open with these specifications. Alternatively, to compare multiple probes to the same target, please check the Add to Compare box located below the cart. This will result in a probe comparison table where the different specifications can be compared. In this example, for PD-L1, as we can see, 2 probes are standard 20 ZZ probes targeting 124 to 1122 base pairs, while the CD274-01 is a 14 ZZ probe targeting 110 to 802 base pairs. Please also note the sense in the probe name indicates the probe is targeting the sequence complementary to the coding strand. This completes our section on identifying and navigating through our catalog probes on our website. I will now make way for Mindy, who will review the custom probe design requirements with us. Please note, for custom probe design, filling a request form is mandatory. Please contact your account executive for this form. Alternatively, you can contact us via support to access this form.
Hello everyone, this is Mindy. Next, I'm going to talk about the custom probe design. If your gene of interest is not listed in our catalog, ACD can design and manufacture new probes for you in about 2 weeks. We term this RNAscope made-to-order target probes or customer probes. We provide a probe design input form for you to specify your research needs and the probe design requirements for the target of interest. First, I will present the probe design input form for you, and then I will show a few popular research applications using RNAscope system and demonstrate how to use the probe design input form to request custom probes for those applications. So the form contains two major parts. The first part showing on this slide is the information we must have for performing a customer probe design. This includes the species of the target gene, the species of your sample, and official gene names, as well as the GenBank accession number or the target sequence. Later, I will also show you how to find the gene information on ncbi. You can also have the option to assign channels if the probe is going to be used for duplex assay or fluorescent multiplex assay. The second part of the form is any additional requirements from you for the customer probe design. For example, any regions in particular you want to target, or any regions you want to avoid in the target region, or other genes with some high similarity or species you do not want to target. You are also encouraged to provide an overview of your experimental plan and any additional comments or requests in the form. So researchers request the customer probe design for a diversity of research applications. Here I'm going to show a few popular research applications using the RNAscope system and show you how to use the probe design input form to communicate with the probe design scientist and request the customer probe for those applications. One of the frequent requests we got is whether we can design a probe for detecting a human gene in mouse without cross-detecting the endogenous mouse gene. And the answer is yes. This image showing on the slide is a simultaneous detection of human and mouse PKM1 gene in a patient-derived xenograft mouse model. The human probe sends the human PKM1 in green exclusively in the human-originated tumor cells, and the mouse probe sends mouse PKM1 in red dots only in the mouse stroma cells. And to achieve this kind of purpose, we need to screen both human and the mouse genome during the probe design to ensure the probe specificity in both species and make sure it doesn't cross-detect the endogenous PKM1 in mouse. So when you request a species-specific probe, we ask you to specify both the gene species and the sample species. If only the species of the target gene is mentioned, for example, if it only says human PKM1 in this form, then the probe will only be screened against the human genome database by default, and the probe might end up have cross-detection to mouse or other species. And we also suggest you to fill in the do not cross-detect other species field for a species-specific probe. Another popular request is virus detection in a host species. Here we are giving you two examples. On the left side is HIV detection in FFPE human lung tissue, and this is the brown assay. And another example is Zika virus, MR766 strain genomic RNA detection in viral cells. So when you request a virus probe, please do not forget to include the species of the host or your sample that's going to be sent. This way we can design the probe not to cross-detect any host RNA. We also ask you to specify the sequence or accession number of this virus strain you are interested in. You can also specify a particular virus gene or a region as target. For example, here we can make a probe targeting within the polyprotein region of Zika virus. We also frequently design customer probes for verifying knockout mice. As shown here, in order to discriminate the wild-type mice and the mice with mouse BMP2 exon 3 deletion, we design a probe targeting the knockout region in exon 3. The probe can hybridize to the wild-type BMP2 mRNA and generate signal. But since there's no binding site in the knockout mRNA, there will be no signal in the knockout mice. And to request the custom probes for distinguishing knockout mouse from wild-type, we ask you to specify the knockout sequence You can either provide the full sequence within the knockout region, or you can give us the coordinate using the RefSeq accession number as a reference. We also encourage you to mention that this is for distinguishing wild type and knockout in the additional comment field at the bottom of the form. So next, I'm going to demonstrate how to retrieve the gene information and target RNA sequence from ncbi for filling the form. And I will use this mouse BMP2 exon 3 probe as an example to demonstrate. So first, we need to go to ncbi gene database following the link shown on the slide. On the ncbi gene page, you can type in the gene name and the species you want to target and click search. In the search result, you can find the genes and species you want to detect. The first column is the official gene name and the entries gene ID. And the second column is the gene description and the species information. We strongly recommend providing the official gene name and the gene ID because they are unique for each of the annotated genes. A common name or an alias name may be used for more than one target, and if a common name is provided, we maybe end up targeting a different gene or sequence other than the gene of interest. So click on the desired gene name and entry gene ID. You will be linked to a full report of this gene. Start with the gene summary. And the gene entry ID can also be found on this page right below the gene name and the official gene symbol. You can click on the ncbi reference sequence on the right-hand side to directly jump to the RefSeq sequence section. Alternatively, you can keep scrolling down to see more details about this mouse BMP2 gene, and then eventually you'll get to the RefSeq section. If you keep scrolling down, you will first see a diagram of the genomic region with different transcription variants and protein products. And on the upper right side, there's another link of ncbi RefSeq sequence, and to click, and you can jump to the RefSeq sequence section. When you get to the ncbi RefSeq sequence section, you can click on the desired RefSeq ID. And when a gene has multiple transcripts, we recommend pick a representative sequence that shares the most common sequence with other variants. And ACD will aim to target the common sequence to detect all the transcript variants, or as many as possible. Once you click the RefSeq ID, it will be directed to the GenBank page of the mouse BMP2 gene. If the goal is to simply design a probe to target the mouse BMP2 RNA, and you can then just record down the RefSeq accession number on the form and stop here. But to target a specific region for differentiating the wild type and knockout animals, you will need to scroll down to find the exon annotation and the sequences to target. Looking into the GenBank annotation, you can locate the third exon, which is the deleted mRNA region in the knockout mouse. The start and end position of mouse BMP2 exon 3 is 1544 to 3566. And by click on the exon symbol, the exon 3 sequence will be highlighted in this page. And there will be a brief summary about the highlighted sequence at the lower right side of the page. And by clicking the FASTA symbol, you will be directed to the FASTA format of the mouse BMP2 exon 3 sequence. And now you can either copy the sequence to clipboard or save it as a FASTA format or any other format offered in the scroll down menu. I also recommend you to double check if this identified region matches the sequence in your knockout mouse construct, because the exon count can be annotated differently depending on the reference sequences or whether the exon bearing the 5' UTR is counted or not in the original design. And also, alternative way to obtain this target of coordinate is to blast the construct sequence against the mouse genome or RNA database.
Thank you, Mindy. We will now quickly recap and summarize the sections discussed during this webinar. In our probe design technology overview section, we learned that the ACD probe design algorithm is validated to select oligos for optimal hybridization at RNAscope assay conditions and minimal cross-hybridization to off-target sequences. For a probe classification and nomenclature, our probes can broadly be classified by assay, platform, and channel. While selecting RNAscope probes, please pay special attention to the probe type, which is your platform, the species name, the gene name, we use the official ncbi gene name, and specifications, if any. When trying to access the probe information on our website, please use the search bar to find the probe of interest by using the official gene symbol, as this gives the most accurate search results. Use the filters bar to refine the results to appropriate platform and species. And always click View Details to ensure that the target region and the probe pairs are appropriate for your use. In the last section, Mindy discussed our custom probe design, the fields that would be required to ensure complete information transfer to ACD. Please make sure to fill those mandatory fields. And additional information may be necessary when designing probes specific to your research application. With this, we will now address a few questions. So one question that we have is related to our probe QC process. Do we perform QC on our RNAscope probes? We perform in silico verifications for RNAscope probes. As we saw earlier, our probe design algorithm selects oligos with compatible melting temperature for optimal hybridization at RNAscope assay conditions and minimal cross-hybridization to off-target sequences. There is a verification procedure following each major step during the probe design to guarantee the accuracy of design. During manufacture, oligosynthesis QC is performed on each oligo probe. And based on our experience and customer feedback, the standard RNAscope probes are very reliable. We have another question which related to the control probe.
Okay, so this question is, do we require control probes to run the assay? And specifically, what is the recommendation for negative control probe? So I understand for conventional RNA in situ, people frequently use the sense probe as a negative control. However, based on our design, we do not recommend a sense probe by default. The reason is sometimes the transcription does occur on the opposite strand, and lots of these events are not even well documented or not annotated. So in this case, the result may be confusing. So the signal you get maybe not just a random assay background, but actual signal. So therefore, we recommend a non-specific bacteria that B as the negative control. So this is the best negative control. And in terms, yeah, so I think that's it.
If you have questions regarding to our custom probe design form and you would like to get started with it, please note this form is available through your regional account executives. If you do not have an assigned executive, please reach out to support@acdbio.com and we would be happy to provide you a copy of this form to get started on your RNAscope probe design.
We got another question. Can we design probes for introns? The answer is yes, and we have experience designing probes targeting intron, and mostly people use those intronic probes for detecting transcription activation or nascent RNA. Yeah, you can fill in the form if you are interested in designing probes for introns.
If you have questions which are related to your specific design needs, please feel free to fill out this form and send it to us so we can perform a feasibility for you That would be the most optimal way for us to get back to you with our responses on whether our probes can be designed for your particular targets of interest. With this, we conclude our webinar for today. Thank you so much for taking the time to attend this session, and we do hope you found this information useful. This webinar will be posted on our website in a few days. And lastly, we're always happy to assist you with any questions or concerns you may have with regards to getting started with or running your RNAscope assays successfully. Please reach us through any of the methods listed here to contact our support scientists for any technical questions. Also, we will be online. Our scientists are answering your questions via chat, so the webinar forum will stay on. However, thank you so much for your time. And online, we will be answering your chat questions for a short while longer. Thank you.